I like it when he laughs at his own jokes.
I know.
Why? Why?
This is just funny.
What's the general flow of PCR that I gave you yesterday?
You have to denature it.
That I would be happy if you knew these. Denaturing.
Anneal.
And you're doing this without looking at your notes, right?
I am.
No.
I am.
Then you have to add more nucleotide.
Extension.
Or you can say that.
So now, my trained monkeys, they can't do it without your notes.
I did.
It's not enough just to know these words in this order here, right?
You need to know what they mean.
I really want you to conceptualize what they're all about, right?
Yes.
We're not about memorizing. Right? So I'll see if you're going to memorize. So this little machine right here, our wonderful thermal cycler, is what we're going to use to denature the DNA, so everything's going to happen right in here. It is. It only cost me $5,000.
Oh, wow.
Okay.
Right. For
Yeah,
something we could
and
have done all
be
weekend
careful
long,
because
you
some
know.
of your 6-hour kids, I came in and they were touching it.
Oh, they were touching it already?
Yeah.
I thought it was 37 degrees to denature.
It is, you know, a touchable thing, isn't it?
It is, not for your 6-hour though.
We're going to set this at 95 degrees Celsius.
And that's its optimal temperature to denature.
What's that thing
You
called?
know it.
What is it called?
Because we got DNA, we got to denature the DNA, right?
What's that called? Zeiszler?
What's this called? Thermal
Yeah.
cycler.
Thermocycle.
Did you get a picture of that?
A thermocycle?
Do you want me to?
Thermal cycler. It's going to go through, here, Abby, here's what it does. It makes everything that we put in those tubes do that.
Go from extension to denature?
Yep.
It does this every time.
Right.
Do it again. That's such a chat.
I'll edit that out later. Yep. Okay, so denaturing. Let's discuss denaturing. Since some of you are obviously confused about the denaturing process.
Hydrogen.
Right? So we have a DNA molecule. In order to denature it, pull the strands apart, we're going to use heat, and guess what that heat is doing?
Denaturing.
That's true, but specifically, what is the heat denaturing?
Breaking the bond.
So it's breaking the bonds between the strands, and which bonds are those?
Hydrogen.
Hydrogen bonds. So you remember that from first semester, yes?
Mm-hmm.
Okay. So then once that's open, then we have all these nucleotides that don't have partners anymore, correct? Yeah.
But I thought you used helicase to separate
Helicase.
the...
That's what happens in your cells. Helicase is used in your cells.
Could we use?
We don't use it here because we don't need to, because we just use heat.
But could we, like, theoretically, like, use helicase?
Possibly. Yeah.
It's
Why
just
would you
quicker
want
this
to
way.
though?
Well, I'm just saying, would it be possible?
Now-
It would probably be harder, but.
Now think about this though. This process, just so you understand, so maybe you can see this from Monsanto's point of view, since I showed you the Food Incorporated point of view.
You keep moving!
I can't help it.
Oh my gosh.
I ate donuts this morning. I'm gonna move around
Here,
and shake
is that
it.
not your face?
Monsanto
Celsius.
and anybody else who does this sort of thing, to put out a new plant that has new genes, we're talking millions of dollars. We're talking 6 to 18 years before a new plant can go out on the market. That's a lot of time, that's a lot of money. You think you want to patent that?
No.
Oh yeah.
Yeah. If you spent that amount of money, you don't want somebody coming in there and taking your profits away. Right. So it's a process that's difficult to do. And we don't want to spend any extra on extra enzymes if we can just heat something up for free, essentially, and then break it apart. It's easy enough to do, right? Yes, indeed. Part 2. Let's go into some detail about the annealing process. So we're going to place those wonderful little pieces of DNA onto the strands. And what do we call those little pieces of DNA? Not polymerase yet.
Polymers.
Polypeptides.
Not the polymers. We need something. We need something on my. Hold that thought, Caesar. We need something on my lawnmower that I'm going to use to get the gas going. Because I'm going to push it 3 times. Right. Exactly. That's why we call it that, because if you remember, our wonderful little DNA strains had a 3' end and a 5' end, correct?
Yes.
And so the same thing goes for the other DNA strand, correct? What would I have down here?
5.
Once we separate, 5. 3' here. Okay, now we can only add bases, at least an enzyme can only add bases to a 3' end. Do we see any 3' end up here?
Mm-hmm. Wait, how do we know that we can only add it to a 3' end?
It's the only thing the enzyme can do.
Okay.
It's the only open little atom for us to add things to.
Can
What's
only-
on the 3' end?
The hydroxyl.
The hydroxyl group. Yeah, I know you guys see the 3' ends up here, but watch. If I start adding DNA to the 3' ends, I'm doing that. Am I making a copy of what's in here?
No, you're just making it longer.
No, I'm just making it longer, all right. Mark's good with the obvious.
So does that change the prime though? Like.
So I have nowhere to add. I have nowhere.
That's stupid.
Where do I add?
So you can't add it
to the already.
I have to find a sequence that's there, right?
Yeah.
So I need little bits of DNA that are going to find this sequence, let's say right here, and base pair with it. Correct?
Right.
Now watch this. This end would happen to be,
5',
and this end.
3'.
So now do I have a 3' end to make copies?
Yes, okay.
Yes, I can go this way now and make a copy. Same thing down here, but do I start here?
No, you start on
I go on
So
the
you
other
always
side.
start
Because whatever's over here is down here, correct?
right.
Since they're anti-parallel. And then I can run in that direction with the other polymerase, correct?
Okay.
Yeah? Yeah? Good with that?
Okay.
Okay. Those are pretty primers, aren't they? Are you starting to see why all the stuff we studied for